Let's begin. So how do you guys like my no screen?
I think it's pretty neat.
That's actually Clark College. Alright, so if you look on the schedule, what we're doing today is Lab 6, Disc Diffusion and Antibiotics. Please go to the Canvas web page and find, I think under week 4 or week 5, the unknown project's instructions and read it. You need to have some understanding before I talk about it. And I'm going to be talking about it on Tuesday of next week. Let me see if I can quickly get there. And I'll show you the page. The network's a little slow today. I don't know if that's my end or the college. Looks like I've got a mail. So there's the Read the Unknown project for next week. And I think it's under the lab. Ooh, I don't have that on for you. I'm glad I looked. Well, now it's on. And you want to look at the unknown instructions. Spring 2020 is the last time we changed it. And I'll be talking about it on Tuesday. But please read through this unknown instructions. You can also, if you want, take a look at the sample journal, the building a dichotomous game plan, the unknown list. But you don't need to know those until you start writing your journal. Let me see what else is off. Week 5 lecture is on. Week 5 lab, let's turn that on. These are all the different tests that you could possibly use to identify your unknown bacteria. Let's turn on week 6 lab. I'll turn on week 7 lab later. All right, any question about any of that? While I'm at it, let me turn on the notes to chapter 4, because we're done with chapter 4. All right, any questions? So what we're doing today is the Lab 6 Disc Diffusion and Antibiotics. But you do need to read the unknown project instructions. You're going to be beginning that project, well, next week, as early as Tuesday, assuming I get my act together and everything sent out in time. But certainly next week we'll be beginning the project, so you need to read about it, and I'll talk about it next week. And you should be well along in your infectious disease project. I'm trying to look to see when that one's due. That is going to be due on May 15th. So in about 2 weeks' time. A little more than 2 weeks' time, but about 2 weeks' time. All right, any question about any of that? If not, I'm closing that down. Let's come on over to the lab. I just upgraded the lab last night. very minor changes. It mainly made it more accessible. And there is a virtual lab for today. I'm not going to give you the most recent one because I want to go through and read from the one I have highlighted. Guess we enable content. So we're doing Lab 6, Disk Diffusion, where you will be evaluating the effectiveness of disinfectants and antiseptics and the evaluation, especially with antibiotics. There's some pre-reading to do before you do the lab. You'll be watching some video clips, and today there is a virtual lab. So upon successful completion of this laboratory, students should be able to identify the type of chemical agent and the active ingredient used in various disinfectants and antiseptics. You should be able to evaluate the effectiveness of disinfectants and or antiseptics using the disk diffusion method. You should be able to determine the mode of action of various antibiotics. You should be able to evaluate the effectiveness of various antibiotics using the disk diffusion method. You should be able to list the variables that influence the antibiotic disk diffusion susceptibility testing. And I guess we can say how to control those variables. And then lastly, be able to define the terms: sterilization, disinfection, disinfectant, antiseptic, bactericidal, bacteriostatic, antibiotic, spread plate, and zone of inhibition. Any questions about any of that? Yep, don't have any water left. All right, so you should know the term sterilization. That's when we apply something that eliminates all viable organisms, including endospores. And when we give you the media in the lab, it's usually been put in the autoclave, which sterilizes the media. Disinfection is the destruction of vegetative cells, not necessarily endospores, particularly when we're using it on an object. Disinfectants or chemical agents apply to inanimate objects. Antiseptics or chemical agents apply to living tissue. For example, if I clean the toilet with bleach, bleach is a disinfectant. But if a cat has a big cut and I apply bleach to the cut, I'm applying an antiseptic. Any question about that? Now, usually you wouldn't use bleach as an antiseptic because it's kind of harsh. And usually antiseptics are less, irritating than disinfectants. Disinfectants tend to be stronger. To evaluate the effectiveness of a chemical agent, the surface of a nutrient agar plate is inoculated with a particular bacteria. Paper discs are saturated with different chemical agents are then applied to the surface of the agar. The chemical agent then diffuses out of the disc and into the agar, establishing a concentration gradient around the disc. You then incubate the plate overnight and then look at the bacterial growth on the disc. If there's a zone of inhibition, an area of no growth, the chemical is at least effective to a particular bacterium to at least a particular degree. Conversely, if there's no zone of inhibition, the chemical can be assumed to be ineffective. And this table shows you some common disinfectants, the general action, and the preferred use. The zone of inhibition size depends only partly upon the sensitivity of the organism to the disinfectant or the antiseptic. Various other factors also affect the zone of size, including the concentration of the chemical, the molecular weight of the agent. Larger agents will diffuse less well in the agar plate, and so larger agents will have a smaller zone of inhibition than a smaller agent if all other things are equal. And then the density of the bacterial inoculum can also affect the size of the zone of inhibition. So let's look at evaluating antibiotic effectiveness. Antibiotics are categorized as bactericidal if they kill the bacteria, or bacteriostatic if they inhibit the growth of the bacteria. So bacteriostatic does not kill the bacteria. It keeps them from growing. When doctors apply a bacteriostatic antibiotic, they're relying on the immune system to assist in the elimination of the infection, because the antibiotic will not eliminate the infection. It'll just keep it from growing. The Kirby-Bayer disc diffusion test is the most common method for testing for antibiotics sensitivity. The results of such testing help physicians in choosing which antibiotic to use when treating a sick patient. Bacteria isolated, put on an agar plate, spread on the agar plate, and then filtered discs impregnated with antibiotics are applied to the surface of the agar. You then allow the plate to grow, for the bacteria to grow, and then look at the plate. Let me get this up a little higher and blow it up. You then look at the plate and you would say, well, this antibiotic is not very effective, and this one here. And you might say that that one doesn't appear to be as effective as that one, but you'd have to do more to determine whether it is sensitive or resistant to the antibiotic than just measuring the diameter of the zone of inhibition. And I'll talk more about that later. But generally, you could say that this one and that antibiotic are not effective, and maybe this one and that one are very effective. Maybe that one too. Any questions about any of that? So you look at the plate after it's incubated for about 18 hours, and then measure the size of the zone of inhibition. You measure the diameter. And some of the discs will have zones of inhibition because within this region, the antibiotic is high enough that the bacteria are either killed or they're preventing from growing. And so there's a zone of inhibition. Any questions about any of that? Okay, hopefully you understand that, because this point is very important. All right, let me shrink this down. The inhibition zone is dependent on several factors. It's dependent on the diffusion rate of the antibiotic. A smaller antibiotic will diffuse greater in the agar, and then you'll see a larger zone of inhibition than a larger antibiotic. It will also depend on the growth rate of the bacterium, the depth of the agar on the plate, and a few other variables. And we have the variables listed in table 6.2. So to do the Kirby-Bayer procedure correctly, you should inoculate the broth using bacteria that came from several colonies isolated on a non-selective media, and the bacteria have been growing for 24 hours or less. You should adjust the broth turbidity, meaning the density of the bacteria, to 0.5 McFarland units, and this is approximately 1 times 10 to the 5th organisms per mil. You should inoculate at room temperature and always use Mueller-Hinton agar plates that are exactly, this is a mistake here, 5 millimeters deep. No more, no less, because the depth of the agar will affect the size of the zone of inhibition. The plate size should be used too. Use narrow, non-toxic swabs to spread the bacteria on the plate. Spread the bacteria everywhere. Put the disc on the plate, and the disc should be at least 24 millimeters apart from each other. No more than 12 discs with 12 different antibiotics per 150 millimeter plate. The antibiotic disc should be added 5 minutes after the bacteria, but not longer than 15 minutes. Incubate in an incubator for 16 to 18 hours at 35 degrees, no longer than 15 minutes after you add the antibiotic disc. And we control the Kirby-Bayer procedure so that regardless of where you are in the world, whoever is doing the test will get similar zones of inhibition from somebody else who does the test someplace else in the world. Any question about any of that? Okay, so after incubation, you then measure the diameter of the zone of inhibitions, and then you determine if the bacteria is sensitive to that antibiotic, intermediate, I'll discuss more about that, or resistant to the antibiotic. And how you do that is you measure in millimeters the diameter of the zone of inhibition. And yes, you do measure the disc too when you're measuring the diameter. And then take that measurement and look it up in a chart of the NCCLS standards. And the NCCLS standards have, for each antibiotic, for each species, if the zone of inhibition is resistant, then for this one, for example, it has to be less than or equal to 20 millimeters. Realize this will change for different species and for different antibiotics. But for penicillin, Staphylococcus aureus, it's resistant if the zone of inhibition is 20 millimeters or less in size. It's sensitive if the zone of inhibition is 29 millimeters or greater in size. And between that, we say is intermediate. Okay? So to determine whether it's sensitive or resistant, you have to measure the diameter and then look up that measurement in a chart, the NCCLS chart of standards. And it'll change for each antibiotic because each antibiotic will have a different molecular weight, and so it'll diffuse at different rates through the agar. Any question about any of that? Of course, know the terms. All right, let me state that the Kirby-Bayer procedure is very good at testing bacteria for antibiotic sensitivity. Why do you think that might be the case?
Because the paper is saturated with different sort of chemicals, and so if they don't grow, right, like bacteria doesn't grow, that kind of implies something may work, right?
Yeah, you got the right idea. The main reason why the Kirby-Bayer test works so well is that the antibiotic is diffusing out of the disc on the agar, but when a patient has an infection and the doctor gives antibiotic, the antibiotic has to diffuse out of the blood and go to the site of infection. So both are dependent on the antibiotic's diffusion. And the Kirby-Bayer test, for example, this one looks like it's very sensitive to the antibiotic. Most of the time, what the Kirby-Bayer test says, we find in the patient's agreement, so that this antibiotic probably will work very well in the patient. And it's because the antibiotic is diffusing in the patient and on the agar plate, so that the Kirby-Bayer test is effectively measuring what we want it to measure. The Kirby-Bayer test doesn't work so well with disinfectants. And that's because when we use a disinfectant, we're not relying on the disinfectant to diffuse through the agar. Usually we put the disinfectant on a surface and then smear it around the surface to cover the entire surface and then let it air dry. Do you see the difference?
Yeah.
So the Kirby-Bayer test does not effectively measure disinfectants because we don't rely on diffusion for disinfectant to work. Alright, so in this lab you're not physically going to perform any of the activities, but you will be responsible for how to interpret the effectiveness of antibiotics using the Kirby-Bayer diffusion test. Watch a video on preparing a spread plate, and then you review the steps for using aseptic technique for preparing a spread plate and getting the bacteria to grow on the plate everywhere, and we call that a lawn of growth. You inoculate much of the agar surface area as possible with a swab, and you do it not just one time. You rotate the plate, rotate the swab, do it a second time, and then rotate the plate, rotate the swab again, and do it a third time. Any question about that? And that's to make sure we get bacteria to grow everywhere. And then procedure 2, evaluate the antibiotic effectiveness. Watch a little video on preparing a culture for Kirby-Bayer diffusion, and then a little video on interpreting the results for Kirby-Bayer diffusion. Then there's a virtual lab for actually doing the Kirby-Bayer diffusion test. And this lab will take a little bit of time to download onto your computer before you run. And I'm sorry, I don't have the instructions here, but I've got the instructions on your copy for what to do. Like, I think you hit the start button then you hit the menu button, and then you hit the steps button, and then if you need to, hit the restart button. All right, and then once you got your measurement, take that measurement and use the standards for the Kirby-Bayer test, and then determine from the NCCLS zone standards whether the zone of inhibition is sensitive to that antibiotic, or you could say susceptible, or resistant to that antibiotic, or intermediate. And then we abbreviate that S for sensitive, I for intermediate, R for resistance. When you're going to be measuring the zone of inhibitions, you have to use the calipers, which are given to you in this picture. And you do not measure from here to there, okay? Because if you do, that's wrong. If you notice, the 0 mark here is not over here. It starts there. So when you measure this, you measure it from here to this 0 on the right hand, I don't know, tooth of the caliper. And then take that diameter, for this one it's 14, and take a look at the chart. Okay? And what bacteria are we doing? E. coli. And I did this one, cephalothen, was 14. So cephalothen for E. coli is resistant. Okay? And you want to record your measurements in this table here. Sorry, my thing is moving. Recorded the zone of inhibition in millimeters, and then give your interpretation, S, I, or R. And you need to look at the chart, and this will be the chart we're using. And then answer the questions in the worksheet, not in the lab manual. And then submit the questions when you're all done. Any questions about what you're doing? If not, I will be on here until 7:30 to answer any questions. If you log off and you have a question, make sure you get back by 7:30. All right? I will see you later.
All right, thank you.
All right.